Hello, today I'm going to be doing a tour around my lab just to show you some of the facilities that we have available. Are you ready to go with me? Okay, let's go. So this is a gas chromatography machine. If you want to analyze the fatty acid composition of any tissue, you just extract your lipids and then you do your transmetallation and then put a sample on the machine. You can get like the different fatty acids that are available in your particular sample. And then we also have an incubator. If you're doing, let's say, cell culture work and you need like 37 degrees to keep your sample, you can put the sample in the incubator. And this is also a biosafety cabinet in case you're doing cell culture work and you need to do it like in a sterile environment. You can do your cell culture work here. And then we do have a chemical storage cabinet in case you want to store your chemicals. Lots of places to store your chemicals. Then this is a pH meter. If you want to, let's say, adjust the pH of your sample to a particular pH, let's say 7.0, you can use this. And then this is a magnetic stirrer. You just need to put like some of this stir bar in a sample, and then you can stir your sample. Especially there are some solvents that are not. There are some solvents that you prepare, or some solutions that you really need some time to get it cleared. Maybe it takes like a couple of minutes for it to get dissolved, so you can use that for that particular dissolving your sample, just stirring it for like a couple of minutes. And then we do also have like a magnetic stirrer, just like a normal vortex, just in case you're working and then you need to vortex your sample. You can if you want to adjust the speed, you can adjust the speed, and then you can stir your sample. Pretty nice. And then we also do have like a rotator in case you want to rotate your sample. Maybe if you are doing like ELISA analysis, you can use this to rotate your sample for a couple of minutes. Depends on the type of assay that you are doing. And then we also have a couple of gases. Let's say you're running gas chromatography. You need the compressed air. You also need helium for your gas chromatography. We have those gases available. So whenever those gases are finished, it's very easy to just replace those gases. And then we have liquid nitrogen. If you're working on a particular assay and you want your samples to be kept frozen and just maintain that structural integrity, you can use the liquid nitrogen. Just pour a little bit in a jar or something, put the sample inside, and I think the temperature is around minus 180 or so, if I can remember, but I know the temperature is really low, so you can freeze something for a very long time. Then we do have water bath, just in case you want to put your sample in the water bath, especially those that do like cell culture and they want to do it for, let's say, a couple of minutes. This magnetic stirrer, it's really nice. You can set it to a particular temperature that you want, let's say, to heat up like your reagents to a certain temperature. It's and then we also have a microcentrifuge. You can maybe you want to do your plasma separation, and you can use it, just set it to the actual temperature, and then the centrifuge, the RPM, or maybe 2000, 5000 G, and then set it to the correct temperature, then you can use it. We also do have a weighing balance. If you want to weigh your let's say you're preparing reagents, you can use this to weigh your samples, and then depends on the particular type of weight or the amount of sample that you're looking for to prepare your sample, you can use this as well. And then we do have, if you are planning to run, let's say, gel electrophoresis or any kind of electrophoresis or Western blots, there are all kinds of equipment and apparatus that are available to do Western blots, and then there are also some reagents used for Western blot. And there are also all kinds of different pipettes, different volumes of pipette. You can use it, and then P20, P200, P1000, depends on the volume of pipette that you were looking for. And then we also have the place where we do all our lipid work. These are lipid lab. There is another cabinet here, and then there is a place where we do all our cabinets where the hood, the film hood actually, where we do all our lipid work. And then there are other reagents as well, and then there are also gases in case you need anything. And then we also have what's it called again, incubator as well, and depends on the kind of work that you want to do. A couple of racks for holding samples, and then there is also this oven that we can use in case you want to heat your sample for some time. And then there is the fridge and the freezer where you can sample. So it's really nice. And then there are places where you have all of your reagents available, and if you can get to clean do the dishes and clean all your reagents, usually available. All your glasses, it's very recommended to make sure all your glasses are cleaned after doing an experiment. And then we also have like a cold room, and just in case you want to put your sample at 4 degrees, you can keep it in cold room for as long as you want, especially for samples that need to be stored at 4 degrees. We also do have a spectrophotometer, just in case you're doing an analytical assay that require you to measure the spectrophotometer at a certain nanometer. You can use this to measure, let's say you're doing triglyceride assay. These are very fantastic to use. And then we also do have, if you're doing real-time PCR, this machine is used for real-time PCR. And then if you want to measure concentration of RNA, you can use this nanodrop to measure the concentration of your RNA or your DNA. Then I haven't used this machine, but I presume it's for gel chemidoc, so I think it's for imaging gel. Let's say you've run gel electrophoresis, or as the case may be, and you just want to visualize the gel after staining. You can use this gel doc to do your imaging. And then this LCMS machine, I will just show you a little. 5 seconds. Okay? 5 seconds. Yes, 5 seconds. Okay, thank you. You can also use this oven to heat your sample. Depends on the particular temperature that you're looking at. Just set the temperature. There is like a thermometer that they can use to see how the temperature is in the just different labs that are on this hallway. Depends on the type of assay and type of analysis that they're doing in the lab. Pretty awesome. Then we also have like a room, in case you want to do like a presentation, and there's a conference room that you can use. You just have to plug your stuff through the computer, and then you get projected on the screen. You can do your practice presentation, and then maybe you're doing it as well. You can also use this for your practice presentation. So that's all for today. I hope I've been able to give you a little brief tour around some of the things. These are just there are some that I don't have the license to. I wasn't given the authority to share, but these are just a few ones. There are a lot more equipment and materials that are available in the lab that I haven't shared today. And I do hope you enjoyed this video. If you enjoyed this video, do well to hit the subscribe button, and then I'll see you in the next video. Thank you.